You're in your car waiting at the traffic light. It glows green and you're glad to zoom away. Similarly, paddy fields could glow green when they're ready to be harvested by a farmer. Wouldn't this be amazing? This isn't science fiction. It could very well be within the realm of possibility. This is possible because of a phenomenon called bioluminescence. Bioluminescence is the production of light within the body of a living organism. It can be seen in different kinds of organisms. For example, the most popular bioluminescent land creature, the firefly. Fireflies use their natural bioluminescent light as a means to find a mate. The formation of their natural beautiful flicker is possible because of a very specific biochemical reaction. The reaction happens within the cellular environment of the firefly. The reaction takes place when the naturally occurring chemical luciferin and the enzyme luciferase combine in the presence of ATP, which is the primary fuel source within the cell. This results in the formation of luciferin adenylate, which then combines with oxygen to give out light. Very little energy produced in this reaction is dissipated as heat. This is why bioluminescent light is called cold light. Biomimicry is a big part of biotechnology. It involves taking inspiration from nature to create innovations that benefit humankind. This charming ability of the firefly to light itself up has encouraged scientists to use this phenomenon in biotechnology. The gene that confers bioluminescence can be used to study gene expression. When a scientist wants to study a gene, they usually do this by inserting that particular gene, which we can call the gene of interest, into an expression vector. An expression vector is a vehicle that ferries the gene of interest into a host cell. Now, having said that, how can a scientist determine if the gene of interest has actually gone into that host cell? This is done by using a reporter gene. A reporter gene is a gene that encodes for a protein that is very easily detectable. For example, the bioluminescent gene or the luciferase gene of the firefly we have been talking about. Hence, we can determine how much of the protein of interest is being produced by the gene of interest by quantifying the amount of bioluminescent light generated by the luciferase gene. Bioluminescence is also useful in cancer research. It is used to determine if a potential cancer drug is effective or not. Cancer drugs work by killing rapidly proliferating cancerous cells. In order to study the effectiveness of a cancer drug, researchers plate cancerous cells in culture wells. As a control, cancer cells are grown under the exact same conditions but are left untreated. The primary cellular energy fuel, ATP, can be produced only by live cells, and ATP is required for the luciferase reaction to take place. Hence, only live cells will be able to give out bioluminescent light. If a cancer drug is effective, it would have killed most of the cancerous cells. The control cells, which were left untreated, will be producing much more bioluminescent light as compared to the treated cells.